Welcome to Week 5 of BIOT 643. This week we'll be discussing RNA technologies. In this week's lectures we'll be discussing RNA detection and its applications. Within this we'll be discussing northern blotting, differential display analysis, subtractive hybridization, and microarrays. The next topic will also be applications of RNA activity, where we'll be talking about aptamers, antisense RNAs, ribozymes, and RNA interference. So first, RNA detection and its applications. So why detect RNA? Well, this is important for the identification of the genes that are expressed at any given time in a cell. So why? Well, basically, the expression of RNA denotes the regulation of gene expression. It allows you to determine if a gene is transcribed in all tissues of an organism or only in certain tissues. It allows you to determine the temporal pattern of expression of individual genes during growth or development. It allows one to determine the physiologic response to a stimulus. And it also allows you to determine if a disease or condition is correlated with a change in gene expression. So the first technique that we'll look at is northern blot hybridization. Northern blot hybridization is the RNA equivalent of southern blotting. RNA molecules are separated via gel electrophoresis. The separated molecules are transferred to a support membrane. A labeled probe is hybridized to the RNA molecules. These probes are short pieces of DNA or RNA labeled with a marker. This marker can be fluorescent or radioactive. And the rationale for this hybridization is the complementation of bases. The labeled probe is then visualized. It's important to note that RNA is very sensitive to degradation by nucleases present in the environment. As a result, all reagents and disposables should be certified as nuclease-free. Here we show you diagrammatically how northern blotting works. You have your RNA is added to the electrophoresis gel. It is electrophoretically separated. It is then placed into a tray. On top of the electrophoresis gel is placed a nitrocellulose membrane. This is placed in contact with the electrophoresis gel. Then paper towel is added to the nitrocellulose membrane. While this is shown as paper towels, in actual practice this is generally filter paper. Again, this is placed on top of the nitrocellulose membrane. Then a weight is added to the paper towel to ensure complete contact between the nitrocellulose membrane and the electrophoresis gel, and a firm contact with the filter paper to make sure that a good seal is achieved. Transfer buffer is then added to the tray, and based on this transfer buffer is then drawn by absorption to the paper towels through the gel and through the nitrocellulose membrane and into the paper towels. This way, any molecules in the gel are transferred to the nitrocellulose paper. The nitrocellulose paper will bind all RNA molecules which it comes in contact with. When it is complete, We remove the weight and the paper towels. And now the nitrocellulose paper is an exact copy of the RNA molecules which were on the gel. Now, for the hybridization, we now put again on a tray. With our probe solution is now added to the tray with our nitrocellulose membrane in the tray. Our labeled probe is added to the probe solution. This is allowed to incubate, and where it finds complementary bases, you get binding of your probe to that piece of RNA. If it's present on the membrane, you will now see binding. Once that has occurred, the membrane is removed and washed, and then it can be visualized. It can be visualized by light or by some other detection mechanism depending upon what your probe is. So then what you see then is wherever your probe binds, you now see, you're able to visualize the presence of the complementary RNA on the gel. So this now is the RNA containing your target. There are certain limitations to Northern blot analysis. Only a few samples can be tested at one time. A large amount of messenger RNA is required, anywhere from 5 to 10 micrograms of RNA. To address these limitations, several techniques have been developed to enable the analysis of several thousand genes simultaneously. These include differential display analysis, subtractive hybridization, and microarrays. First, we'll present differential display analysis. Differential display analysis involves the separation and cloning of individual messenger RNAs by means of PCR. The next step is RT-PCR. Partial cDNA sequences are amplified from subsets of messenger RNA. For primers, differential display analysis uses one primer that is anchored to the poly A tail of the messenger RNA. The other is a short, arbitrary primer that anneals at different positions relative to the first primer. The diagram at the bottom shows the example of the messenger RNA with the poly A tail. Reverse transcriptase results in a cDNA with a poly T tail. The initial amplified cDNA molecules are further amplified by standard PCR, as shown in this diagram. Subsequently, gel electrophoresis enables comparison of gene expression levels. To show you how differential display analysis works, experimental sample 1, experimental sample 2. The total RNA population is isolated in solution. Using RT-PCR, as we described previously, with where one primer targets the poly A tail of messenger RNA, the messenger RNAs are specifically amplified using the RT-PCR, so you now have cDNA isolated from the messenger RNA. This messenger RNA is then amplified using standard PCR. The resulting amplicons are then separated by gel electrophoresis. And here we show an example. In this case, you see a set of messenger RNAs which are upregulated in sample 2, a set of messenger RNAs which are expressed only in sample 2, versus a set of messenger RNAs which are expressed only in sample 1. These specific bands can be removed from the gels and sequenced to define what these specific messenger RNAs encode for. Here is another example of how differential display analysis can work in a normal cell isolated messenger RNA versus disease cell or disease condition. Again, you see in the first arrow points out a messenger RNA which is expressed only in the disease condition. The second arrow shows a messenger RNA which is overexpressed in the disease condition versus the lower arrow where you have a messenger RNA which is overexpressed in the normal condition. In the B sample, we show a similar as an actual gel where you can see certain bands which are overexpressed in certain conditions and certain bands which are underexpressed. The next technique that we'll discuss is subtractive hybridization. Within subtractive hybridization, the messenger RNA is isolated from cells. Your sample, the test or tester or test sample, the control or driver. The messenger RNA is converted to cDNA via RT-PCR. Adapter molecules are attached to both cDNA populations. The tester or sample cDNAs are incubated with the driver cDNAs. The common complementary molecules bind to each other. Primers specific to each adapter allows amplification of only the uniquely expressed genes. The disadvantage of this is it's technically very challenging. In addition to being technically challenging, the remaining differentially expressed cDNAs are still unknown, and they must be isolated from both the tester and the driver and analyzed by other methods. Both differential display analysis and subtractive hybridization have largely been supplanted by microarrays. So let's talk about microarrays. So for microarray chips, it utilizes DNA as the target. Thousands of known genes are added onto a tiny microarray chip in known locations. And the target spots can represent the entire set of known genes from a sample. Unlike subtractive hybridization, the entire set of transcribed genes, or transcriptome, can be tested at once. So here, for example, we have experimental sample 1 and experimental sample 2 again, and the total RNA populations. And as previously, we can use RT-PCR to pull out or amplify the messenger RNA in both experimental samples. In this case, we can use fluorescent probes of different colors in each of our two samples. The cDNA from each sample is labeled with a different fluorophore. For hybridization to the microarray targets, here we have, for example, our experimental sample 1, which is labeled with a red fluorophore. Our experimental sample 2, the cDNA, is labeled with a green fluorophore. And these are added to our arrays. These are then exposed to the appropriate UV light, and then what we see is wherever there's expression, we see the intensity of, if the sample, say sample 1, the normal is expressed and it's present, it turns red. If it's not, it stays unstained. The same is true in our sample 2, which has the green label. When the samples are present, we see a green label. When the sample is absent, we see, in this case, a clear. Looking at the samples that we have here in our experimental condition, gene A doesn't appear to be expressed in the normal sample, while it's expressed in our experimental sample 2, the disease state. So gene A is amplified in the disease state, or upregulated. Gene 2, or B, is expressed in both, so it's equally expressed. Again, gene C is expressed in both, so it's equal, whereas gene D is upregulated in the disease state. Gene E is equal in both the normal and the disease state, whereas gene F is downregulated in the disease state. Since the identity of the gene in each spot is known, we can then say what genes are specifically upregulated and downregulated or equally expressed in the disease condition. So we can use these microarrays for different applications, such as comparative genomic hybridization. This allows us to classify tumors, for risk assessment, and to predict prognosis. It can be used for expression analysis, to enable drug development, to predict drug responses, and to allow us to develop unique or targeted therapies. These microarrays can also be used for mutation or polymorphism analysis. Again, this enables specific targeted drug development, as well as the tracking of disease progression. The next technology that we'll present is gene silencing using RNA. Here we show an example of, normally what you see is you have DNA. The DNA is transcribed to give you your messenger RNA, and the messenger RNA is then translated to give you a protein. However, one can use things such as an antisense oligonucleotide to block translation. You can use a ribozyme or a DNA enzyme, again, which will block translation by degrading the messenger RNA, or small interfering RNA, again, which degrades or blocks the translation, ending in no protein translation. So this, even though you have the messenger RNA transcribed, there is no translation to the finished protein. So among the gene silencing RNAs, the first one we'll discuss are aptamers. Aptamers are synthetic oligonucleotides, roughly 15 to 40 base pairs in length. These are designed to bind to proteins that normally do not bind to nucleic acids. Aptamers have organized secondary and tertiary structures, and they bind to specific proteins with very high affinity. The binding of the aptamer prevents the protein from exerting its effect, but the proteins are still expressed normally. Here we show The folded RNA aptamers with the 5' and 3' flanking regions binding to a target molecule and resulting in the aptamer target complex. The next is gene silencing RNA technology is antisense RNAs. The translation of a messenger RNA can be prevented by providing RNA or DNA molecules that are complementary to the messenger RNA. Antisense RNAs can be used to suppress or reduce the expression of a protein known to be involved in a disease. The major advantage of using antisense RNAs are its sensitivity and specificities. Antisense RNA technology has been used as the basis for various drugs, both in cancer, viral infections, and inflammatory diseases. For an example, Vichavine has been used for CMV-induced retinitis in AIDS patients, and it was approved by the FDA in 1998. So for antisense RNA technologies, short RNA sequences that are complementary to the target gene are used. The antisense oligonucleotide must be introduced into the cell of the organism for the effect to take place. Here we show an example where you have the antisense oligonucleotide being added to the cell. On the right side of this diagram shows the normal series of events where you have the DNA, which is transcribed to generate the primary transcript. It undergoes RNA processing to result in the messenger RNA. This messenger RNA can then bind the antisense oligonucleotide. The binding of this oligonucleotide prevents the translation of the message, so there is thus no translation of the resulting protein. So what is the mechanism of this antisense activity? There are two mechanisms of action. One is the activation of the ribonuclease RNase H. And that's shown in diagram A, where the RNase H is cleavage. You have the messenger RNA, you have the antisense oligonucleotide bound to the messenger RNA, which activates the RNase H, which actually cleaves the messenger RNA. Or there can be a physical blockage of the ribosomal access to the gene. So you have the messenger RNA, your antisense oligonucleotide binds to the messenger RNA, so when the ribosome starts translating the messenger RNA, it's physically blocked from its movement down and translation of the messenger RNA. So what are the limitations of this antisense technology? Well, first is the ability to internalize the oligonucleotides. It's one of the things we discussed. In order to have its mode of action, this antisense oligonucleotide must be introduced inside of the cell. So that is one limitation. Its stability, its effectiveness. These generally are termed as knockdown and not completely knockout technologies, as well as off-target effects. These are small oligonucleotides, so they may have binding other than your specific target of interest. So for the next applications of RNA activity, we'll talk about ribozymes. Ribozymes are ribonucleotide enzymes, so it's a derivation of that term. Unlike most RNA molecules, ribozymes possess catalytic activity. ribozymes bind to a specific complementary sequence on a messenger RNA molecule and cut it. Ribozymes have two domains: the RNA substrate binding domain and the catalytic domain. The ribozyme substrate binding domain recognizes a specific sequence that recognizes and hybridizes to a complementary sequence on the target RNA. For the catalytic domain, this domain actually cuts the RNA at the specific target site. Here, these are shown in this diagram. This is the substrate binding domain and the catalytic domain. Next, we'll talk about RNA interference, or RNAi. Small interfering RNAs, or siRNAs, trigger the process of RNA interference. Small interfering RNAs form a complex with RNA, protein, and a nuclease to form an RNA-induced silencing complex, or RISC. The mechanism of action is double-stranded siRNA binds to the RISC, then unwinding of the double-stranded siRNA, followed by recognition of the target sequence, the specific messenger RNA. The target messenger RNA is then cleaved by the RISC, preventing any translation of the gene. Here, we show this diagrammatically. The small interfering RNA, a 21 to 25 base pairs RNA strand, is targeted to a specific gene. B, within cells, the siRNA unwinds and is incorporated into the RISC, a stable protein RNA complex. The siRNA is directed to a target messenger RNA that is known to be involved in a diseased pathway. Then D, the messenger RNA undergoes degradation, thereby interrupting the protein synthesis of the targeted gene. Here we show the therapeutic mechanism of RNA interference. So we start with A. The short interfering RNA is designed to correspond to the gene target. B. The siRNA synthesized with drug-like properties. It has stability and conjugation for delivery. C. The modified siRNAs penetrate the cell membrane and harness the RNA interference mechanism for gene silencing. Followed by D, gene silencing achieves a therapeutic effect. Animation showing the mechanism of RNA interference can be viewed clicking on the link shown below within the reference slide deck. So some of the RNA interference applications are reverse genetics. The known sequence of a gene can be used to find its function by inhibiting its activity in vivo. siRNA can be created to degrade the messenger RNA from a specific gene, and the effects of the gene knockout can then be studied. Or therapeutics, the siRNAs to a disease-specific messenger RNA can be delivered as a treatment for the disease. So, what are the advantages and limitations of RNA interference? Well, the advantages are it is highly specific. The double-stranded RNA is created to target a specific gene. The systemic response, the double-stranded RNA can move through the organism. The response is not just localized to the site where the siRNA, the double-stranded RNA, is introduced. And it works across a wide range of species and disease. So what are the limitations? siRNA design determines its efficiency. The high specificity is required to reduce off-target effects. And delivery into the cell can be difficult. It can require lipid-mediated transfection or electroporation or microinjection. So for your review, these are the references that were used.